座長は宮崎大学解剖学校の長期形態科学分野の、豊島先生におっしゃいます。
Localization of morphology may be described mitochondrial elongation factor 1 MITF1 to extend MCF7 発表お願いします。
皆さん、大学から出演です。よろしくお願いします。今から英語で話します。Mitochondria are critical in many cellular processes such as metabolic homeostasis, steroid synthesis, calcium homeostasis, energy production, antioxidation, and apoptosis. Mitochondria are dynamic organelles, which means their morphology can be changed by balancing between fission, fragmentation, and fusion elongation. Mitochondrial fission is important for the cell division. During the mitochondrial fission, cytochrome C oxidase release, and it enhances the protein caspase cascade and induce the apoptosis. Mitochondrial fusion induces the ATP production and resulting in cell proliferation and cell survival. In the mitochondrial fission, DRB1 is the major fission-related protein which located in cytoplasm. When there is stimulus, DRB1 was phosphorylated. This phosphorylated DRB1 is the active form, and that active form is recruited by the FIX1 protein. FIX1 protein located at the outer mitochondrial membrane. So this phosphorylated DRB1 protein occur oligomerization and perform scissioning and resulting in the mitochondrial fission. Recently, MIEF1, new mitochondrial fission-related protein was discovered. It is located at the mitochondrial outer membrane. In case of MIEF1, it can bind to the inactive form of DRB1. So this binding inhibits the phosphorylation of DRB1, and it also inhibits the fission actions of the DRB1. So this imbalance event leads to the mitochondrial elongation. And estrogen is very important for the cellular expansion. When estrogen enters into the cells, estrogen receptor alpha and beta carry the estrogen and bind to the ERE receptor of the ERE binding sites of the estrogen receptor related genes, and it induces the transcriptions of estrogen receptor related genes and resulting in the many cellular function. It is reported that estrogen can regulate the mitochondrial morphology. However, the prescribed mechanism is unknown. So in this study, we emphasize on the possible role of MIEF1 in mitochondrial localizations and morphology by estrogen. In this study, estrogen responsive MCF7 human breast cancer cell line was used. MCF7 cells were treated with different concentrations of estrogen for 12 hours. And then RT-PCR results showed that mRNA of the MIEF1 was obviously increased after 10 nanomolar estrogen treatment. Western blot result also showed the similar result. After that, the effect of estrogen with or without ICI were checked. ICI is the estrogen receptor antagonist. After 12-hour treatment, MIEF1 estrogen treatment. However, this increased expression was suppressed by ICI plus E2 treatment. And Western blot results show similar. So from these results, it reveals that estrogen upregulates the MIDF1 expression through estrogen receptors. So to confirm that finding, we plan to use another breast cancer cell line with different estrogen receptor expression. And also we plan to use siRNA, ER alpha and siRNA, ER beta development. After that, the localizations of MIDF1 in MCF7 was checked by immunohistochemistry. In this experiment, OS-phosphide is used as the mitochondrial marker. OS-phosphide is located at the inner mitochondrial membrane and it involved in the ATP protection. So in this figure, MIDF1 expression show the small dotted pattern, and also the expression is moderately expressed. And these MIDF1 and OS-phosphide protein co-localize, partially co-localized. After estrogen treatment, MIDF1 expression intensity increased, and also the expression pattern changed from the fine dotted pattern to the large specular pattern. So from this finding, it is possible that estrogen can change the mitochondrial morphology from small dotted pattern to large specular pattern. To demonstrate the mitochondrial morphological changes, we plan to do immunoelectron microscope later on. As summary, estrogen and its receptor regulate the expressions of MIDF1 in MCF7 breast cancer cells. Estrogen effect on staining pattern changes of MIDF1 from moderate stain, small dot to strongly stained, large closed pattern. It is suggested that estrogen regulates the mitochondrial morphology. As a conclusion, MIDF1 is the one possible candidate which involved in the estrogen-induced mitochondrial morphological changes. Thank you so much. Thank you so much for your